Unlike autotrophs, which are organisms that synthesize nutrients through photosynthesis, heterotrophs are living things that must feed on others, such as plants or animals, because they cannot provide their own energy supply by photosynthesis. The carbohydrates, lipids, and proteins made originally by autotrophs are consumed by heterotrophs, and then broken down into smaller units, usually single or simple molecules, that they can be easily absorbed. Heterotrophic organisms include that of animals, fungi, and most bacteria. A heterotrophic animal eats plants or other animals which have eaten plants, or herbivorous animals, thereby making them carnivorous. Heterotrophic processes in animals. In heterotrophs, obtaining nutrients and energy involves several steps. This is exemplified within the human digestive system. Ingestion, the intake of complex organic food, also known as eating. Digestion, the breakdown of chunks of food into smaller, simpler, and soluble molecules that can easily be absorbed. Physical digestion is predominantly through the chewing of food, but may also be broken down by the churning action of the muscular stomach wall. The aim of digestion is to obtain molecules small enough for absorption. The product of this includes amino acids, which are proteins, simple sugars, which are carbohydrates, fatty acids or glycerol, which are lipids. Absorption, where the basic units of life, an example, glucose, amino acids, fatty acid, and glycerol, are small enough to be absorbed across the wall of the digestive tract, and either into the bloodstream or directly into the cells. Assimilation, the building up of end products into useful substances, either as new biological material or as an energy source. For example, in mammals such as humans, blood transports the food molecules to where they are needed in the body, and can be reassembled by cells into one of two things. Structural parts, an example, lipids and proteins that form a structural part of cell membranes. Protein fibers in muscle tissues are also another example. Energy storage, an example, fatty tissue or fat beneath the skin, glycogen as a form of animal starch, or protein that cannot be stored. Digestion, the elimination of undigested food as waste. For example, fiber such as cellulose, insoluble organic carbohydrates, that is the main component of cell walls in plants. Cells that cannot be digested by humans act as roughage, passing through the digestive tract and out of the anus. The process of digestion is not to be confused with excretion, which is the elimination of chemical waste products of metabolism. That is, excretion involves chemical wastes that are actually made by the body. For example, urea excreted in urine. Teeth and surface area. Most vertebrates that have jaws and teeth that enable them to obtain and process foods. This includes fish, amphibians, reptiles, and mammals. Mammals in particular involve two stages of digestion. Mechanical or physical breakdown, where chunks of food are physically broken down by chewing. Chemical breakdown, where the digestive enzymes act on the food to chemically break down the large complex molecules into simpler and smaller molecules. These enzymes are most efficiently digested when the food to which they are exposed has a large surface area to volume ratio. This in turn speeds up the rate of reaction. Through this, the role of teeth is seen.
This is the Koch and Mentos experiment to simulate digestive enzymes acting on food in relation to the surface area to volume ratio of said food and the rate of reaction involved. To perform this, we have different forms of the Mentos, two of them in each to heighten the reaction with Koch and Mentos. As you have here, the whole, the corded, and the powdered. The Koch is to simulate digestive enzymes and the chemicals involved in breaking down the food. The utensils involved include a teaspoon to stir, the Mentos, 3 Diet Cokes in 3 separate cups, all the same, a measuring device of milliliters, The Diet Coke can, now empty. The Mentos as it's here in different forms, a hammer to powder, a knife to quarter, and the whole was left alone by itself. And lastly, we have a stopwatch to measure the time taken in the breakdown of the food, or rather the Mentos. I will now put the Mentos each at separate times into the Coke and record it on the stopwatch and stir it consistently until the reaction involved between Coke and Mentos ceases. The Coke with whole Mentos will now be inserted. And it is timed. Now the corded will be inserted. Lastly, the powdered or crushed.
The rate of reaction is the speed at which a chemical reaction occurs. If a reaction has a low rate, that means the molecules combine at a slower speed than a reaction with a high rate. Some reactions take hundreds, maybe even millions of years, while others can happen in less than 1 second. Our analogy is carbonization. Remains of deceased animals can take millions of years to form into fossils. The time varies depending on the temperature, concentration, and pressure placed upon these remains. If there is a greater amount of these effects taking place, the faster the remains will become fossils. This is an example of stress factors placed upon chemicals, causing the rate of reaction to change. The larger the amount of stress factors placed on molecules, the faster they react. In carbonization, the pressure placed upon animal and plant remains from the earth compresses the remains, causing a denser area in which the molecules bounce around. Molecules bounce around because of the temperature placed upon them from being in close proximity to the Earth's core. The higher temperature increases the amount of energy inside these molecules. With the experiment done in class, the same principles apply. The larger the surface area exposed, the quicker the tablets reacted.